Okay, in this video we're going to talk about the history of and the research that has led us to our discovery that DNA was the genetic material. So here's some of the different people that led to our understanding of DNA as the genetic material, the structure and function of DNA, and the replication of DNA. And we're going to talk about each of these just a little bit. So the first person that we want to talk about is Thomas Hunt Morgan. So Thomas Hunt Morgan was studying fruit flies, and he noticed that there was a certain variant, a white-eyed mutant, that was only male in his population. So he began to wonder why that was. So like I said, he was working with Drosophila, or fruit flies, and based on his study of fruit flies, he associated this particular phenotype, the white eyes, with a specific chromosome. He noticed that the white-eyed males had a specific X chromosome different from the red-eyed males. So he was the first person to relate the idea or to come up with the idea that chromosomes are where our genetic information was stored. So Morgan's conclusions: genes, the things that control our traits, are on chromosomes. But we still weren't sure, was it the proteins in the chromosome or was it the DNA in the chromosomes that actually made up the genes? Initially, most scientists thought that proteins were the genetic material. And why would they think that? Well, if you look at the structure of DNA, it's pretty simplistic. But if you look at the structure of proteins, it's very complex. So they basically thought like this. Proteins must be the genetic material because their structure is so much more complex than DNA. And then comes the next step in our understanding and leading us to this idea that DNA, not protein, was the genetic material. And that was discovering what was called the transforming principle. So Frederick Griffith was studying Staphylococcus bacteria that cause pneumonia. And there are different strains. Some staph bacteria cause pneumonia. They have a rough appearance, and some don't. They have a smooth appearance. So he was trying to find this cure for pneumonia. So the harmless bacteria, or the rough bacteria, and he mixed them with heat-killed smooth bacteria. And what happened was pretty surprising. He took two bacteria that by themselves shouldn't kill the mice, living harmless and dead harmful bacteria. But when he mixed those two together, the mouse died. And also he discovered when he looked at the blood of the dead mouse that there were live pathogenic harmful bacteria. So something was changing the bacteria from harmless to harmful, and called that the transforming principle. So this substance was passed from the dead bacteria to the live bacteria to change their phenotype. But he didn't know what it was. But he called it the transforming principle. So here's an experiment. So there's basically 4 steps to an experiment. Experiment 1, he took live pathogenic smooth bacteria and injected those in the mouse, and the mouse developed pneumonia and they died. And he took some live non-pathogenic rough bacteria, injected those in the mouse, and the mouse lived. Again, not earth-shattering. Took heat-killed pathogenic bacteria, injected those into the mouse. The mouse lived because the bacteria were dead. They weren't able to reproduce and cause the disease. But then, this is where the confusing part came. He mixed heat-killed pathogenic bacteria from C and the non-pathogenic bacteria from B, where separately the mouse would live. And the mixture, though, killed them, and the mouse died. So again, and we saw living harmful bacteria in the bloodstream of the dead mouse. So transformation is a change in phenotype. Something in the heat-killed bacteria could still transmit disease-causing properties to the living, harmless, or non-pathogenic bacteria. Again, doesn't know what it is. Could be protein, could be DNA, not sure. So Avery, MacLeod, McCarty then decided they're going to try to determine whether it was DNA or protein that was the transforming principle. Now, the their working hypothesis was that protein should have been the transforming principle. So what they did in their experiment, they took and they purified both DNA and protein from the Staphylococcus pneumonia bacteria, the harmful bacteria. So basically, they took the chromosomes out, and in one tube they would add enzymes that would break down all the proteins that would give you the purified DNA. In another tube, they would put enzymes that break down DNA to give you the purified protein. So when they injected the purified protein into the bacteria, nothing happened. And that was kind of a shock. And then they injected the DNA, and the mouse would die, and we saw transformation of the harmless bacteria into the harmful or virulent pathogenic bacteria, and the mouse died. So this was kind of the first evidence that DNA was the genetic material. And then another study was done by Hershey and Chase, and they set about to confirm the results of Avery, McLeod, McCarthy's, but they did it using bacteriophages, which are viruses that infect bacteria. And this is what we call the classic blender experiment because there actually is a blender involved. And you'll see that here in just a minute. So bacteriophages are viruses that can infect bacteria. So they set about, and they knew a bacteriophage was very simple. There was protein coat and there was DNA. So we weren't sure what got into the cell because we weren't able to see this process at this point in history. And they grew the viruses in two media that were radioactively labeled so they could follow and see whether the protein or the DNA entered the cell when the cell was infected. So they radioactively labeled the proteins with radioactive sulfur, which is a common component of protein, and they radioactively labeled DNA with phosphorus, which is in DNA. So they grew the phages in this radioactively labeled media. They picked up either the radiation in their protein or in their DNA, depending on which part of the experiment they were doing. And then they allowed those cells to infect the cells, and then they looked to see where did the DNA end up. So this just kind of shows the process. So again, you have on the left side, you have radioactive protein. On the right side, you have DNA. Allow them to infect the cells. They agitated them by actually putting them into a blender to break the bacteriophages away from the cell. And what they found was the radioactivity stayed outside the cells in the protein labeled experiment, but was inside the infected cells in the DNA labeled part of the experiment. This is just another picture showing the process here. So this was confirmation that DNA was the transforming principle. Here's Hershey and Chase.